Welcome again friends, welcome again in another video lecture of our recombinant DNA technology video lecture series. So in this video we are going to talk about restriction endonuclease as it was promised. Okay, so what restriction endonuclease is? Let's start without any wasting time. Okay, so restriction enzyme, or you can say restriction endonuclease. Okay, so what they are, suppose this is a bigger chromosomes. Okay, and you need to isolate a small portion from it that I have mentioned here in red color. Okay, so by using restriction enzyme you can cut this double stranded DNA into fragment. Okay, so what is the, for doing this there is a requirement for restriction enzyme. The requirement is restriction endonuclease will act, again repeat, enzyme restriction endonuclease will act only on those we can say nucleotide sequences that are recognized by them. Okay, so what restriction endonuclease first that will recognize a nucleotide sequences that will be specific for that particular enzymes. Okay, so what they are? So restriction endonuclease are simply the enzyme that have the ability to cut the double stranded DNA into fragments after recognizing that I am telling about specific nucleotide sequences known as restriction site or recognition site. That means restriction enzyme, when you will add this restriction enzyme along with this bigger chromosome to cut a target DNA, restriction endonuclease will know the specific nucleotide sequences of that target DNA and it will cut up this into fragment. That is the overall idea. Okay, so it was generally isolated, it is generally isolated from bacteria. So why they are mostly found in bacteria? It was thought to be evolved to resist the viral attack in case of bacteria, because we know, we all know that bacteriophage mainly have the ability to attack bacteria and which can be detrimental for bacteria. So by with the help of evolution they have evolved this kind of enzyme that have the ability to resist the entry of viral DNA into the bacteria. Okay, so let's talk about their discovery first. Okay, so first it was first the existence of something like restriction endonuclease was first given by W. Arber. He found that when bacteriophage try to enter its DNA into host bacterium, there is something in host bacterium that have the ability to cut up this cut up this DNA, or we can say viral DNA, into fragments and resists its entry. Okay, that means host bacteria have something that have the ability to resist the entry of viral DNA into its own body, we can say, or it have the ability to prevent the process called transduction. Okay, so but he do not ever have but he cannot isolate that particular product. Later in 1970, Hamilton Smith along with its co-workers first isolate restriction endonuclease from Haemophilus influenza species. The strain for that particular bacterium was RD. Okay, and the enzyme that was isolated was HIN2. Why and how it was named, we are going to talk about after some time. Okay, now as I have told you, this restriction enzyme have the ability to recognize a specific nucleotide sequences among a large one, right? So we need to talk about the characteristics of that particular sequences that was recognized by HIN2. So HIN2 have the ability to recognize a 6 base pair of double stranded DNA sequence, and we will going to talk about this recognition site or recognition sequence in our upcoming video. You don't need to bother about that. This is just a introductory video. We will going to talk about this in our upcoming videos. Okay, so and the next one is that was isolated just after HIN2 was EcoR1, and it was isolated from Escherichia coli species the strain for that particular bacterium was RY13. Okay. So what we have learned? We have simply learned that recombinant restriction endonuclease are the enzymes that have the ability to cut a specific double stranded DNA that was recognized by them into small fragments. Okay. And these specific recognized sequences are called as restriction site or recognition site. Why back, and it was usually found in bacteria. And why bacteria they develop the such kind of enzyme? Simply because they want to resist the entry of viral DNA into bacterium. Okay. Now let's move to the nomenclature. So I am going to talk about with about this with some example. If you want to write the rules, then you can. These are the 1 2 3 4 rules that you need to follow when you are going to nomenclature a particular restriction endonuclease. So let's talk about this. We all know that this is, if it is a scientific name of a bacterium, then the first word simply telling us about genus, and the second one simply telling us about species, and the last one telling us about strain. Okay. So the rule is the first word of the restriction endonuclease will telling us about the genus. So if we want to nomenclature a restriction endonuclease that was isolated from Haemophilus influenza RD, then we need to pick out H for genus, and second and third word will tell us about the species, then I and N will simply tell us about the species, and the fourth word will simply tell us about the strain. Now there is a funda that there is another restriction endonuclease that was isolated from Haemophilus influenza, and strain was in that case RF. So if we pick R for strain, so we also have to pick R for them or two, and it could be a problem, then we cannot distinguish about them. That's why instead of picking R, we will go for D. So hint, and as you have seen here that there is a Roman number. This Roman number simply tells us the order in which enzyme was isolated from that particular bacterium. That means this hint 2 is the second enzyme that was isolated from that particular bacterium. Okay, there was something that was isolated previously from hint 2. Okay, that's why we have assigned number 2 for hint. So first word simply tell us about the genus from which it is isolated, second two words simply tell us about the species, and fourth word simply tell us about the strain, and the Roman numbers simply tell us about the order of the enzyme that was isolated from that particular bacterium. Okay. So by following those rules, let's try to nomenclature an enzyme restriction endonuclease isolated from Haemophilus influenza RF. So we need to pick out H for for our first word that will tell us about genus. Then we will pick IN for indicating the species. Then we will pick F as there is R and there is R. So for avoiding misconception, we will take F. Then we will take 1 because the restriction endonuclease was isolated from that particular bacterium was the first one. Okay. Now let's move. Now we will don't see this first. Okay, let's try to do it on your own. So we are going to assign a name for a restriction endonuclease that was isolated from E. coli. RY13 was the strain, and it was the first enzyme that was isolated from that particular bacterium. Okay, then we will pick the E, which will tell us about genus. We will pick CO, that will tell us simply about the species. Then we will pick R, that will tell us about the strain, and then 1. This one will simply tell us about the that it was the first enzyme that was isolated from that particular bacterium. Okay, that is how a particular restriction endonuclease was named. So restriction endonuclease, again let's revise that restriction endonuclease is simply an enzyme that was in general isolated from bacterium, and they have the ability to cut a specific double stranded DNA into fragment after it was recognized by them. That means this particular site will be considered as restriction site or restriction, or you can say recognition site. Okay. So every restriction endonuclease have the ability to recognize a particular site from a particular nucleotide sequence, and they have the ability to cut them, and that is the idea that we have used in our DNA cloning, okay, for isolating a particular target DNA that we need to that we want to amplify for our further study, or you can say production. Okay. So I hope that the first video regarding restriction endonuclease is clear to you, and if it is, please like the video, share with your friends and subscribe my channel. Thank you. In our next video we are going to talk about blunt end and sticky end, so don't miss it. Okay, bye bye.